this would be funny if it wasn't so silly. I just ran the whole video. I just did my whole presentation with the video with the camera off. That was great. I did set all this stuff up and forgot to push the button. Very good. That's okay, because the first few seconds of my videos don't tend to get shown. They don't show up on YouTube for some reason. They seem to chop off the front, maybe the end, I don't know. But so it's good that I have a couple of seconds here of something that is irrelevant to the process. As usual, my name is George Parris. I'm a PhD chemist, not a biologist. I have some biological ideas and hypotheses, and I've been working here for, I guess this is the 4th or 5th video I've done, trying to summarize for you my ideas, some of my ideas, and how they work together. I'm working towards explaining to you a hypothesis I have, an overarching hypothesis, which I call the Master Developmental Program. And at this point, today, I'm going to talk about differentiation and how that affects development. All right. Before I can do that, I want to introduce you to some terminology and some ideas that if you're a biologist, you know this. I'm sorry for dragging you through it. I do have some personal insights which I think may be interesting and different than you may have heard in your biology classes. But once I get through that, then the new part, the novel part, the stuff that is particularly relevant, is actually going to be fairly short. So I'm not quite sure how long this video is going to last, but we'll go there. I would refer you back to the previous videos I've done, especially the most recent one. I forget the name of it. I don't, by the way, these are not productions. These are lectures. I'm not a guy who edits and manipulates videos. I'm not doing this for money. I'm doing this because I like to teach, and I would like to have a video version of my books. I have written these books. Some of them are over 10 years old, and they are available. Most of them are available on Amazon.com. There's also published articles, and you can find them in PubMed, and you can find them on, there's a couple of other journals that are online. If you search my name, George E. Parris, you will start finding these videos on the internet, on Google, or whatever search engine you may use. Okay, with no more introduction of that, I'd like to go back a little bit and talk about, you remember I've talked a lot about the history of these things. And I think we have two major biases built into our biological systems, our biological perspectives, what we consider to be important, at least what the biologists put into their textbooks. One is, there was a lot of what we look at and talk about was dominated by the introduction of light microscopy. This would be stained cells and focus, good microscopes. About 1900 is when a lot of that came about. And you remember that we're using light microscopes, and we can see things down to about the wavelength of light, which is about 350 nanometers. Okay, if it's smaller than that, you just don't see it with a light microscope. If it's bigger than that, then you can start seeing it, especially if it's properly stained with a good dye. The other thing that we're dominated by, and this is more of a recent event, actually the whole idea of a gene coming in the 1920s, I guess it was. We're dominated by the idea of what we call the central dogma of biology, which leads to proteins. Okay, so really there's an over-attendance on proteins. Fortunately, the idea of RNA and some of the things that RNA does, and the fact that they're transcripts of DNA which produce RNA, which is not coding, doesn't code for proteins. This would be non-coding RNA. This is becoming important, and people are starting to understand it, and it's becoming, I think it will be the key to understanding development as we go down the line. All right. Having said all that, let me get into what a lot of you will consider to be background material. And again, I will be emphasizing pieces which I think will set up and help introduce down the road, the next video or two, the hypothesis I'm trying to explain. Now, as I said, this is a lecture and this is not a production, so you can expect almost anything to happen. If the doorbell rings or something, it will ring. So we'll go. Well, first, let me just bring up a typical chromosome, get you some terminology here. You'll notice on what is my right, your left, I guess, there's a photograph, a micrograph of a real chromosome. This is probably, I think it's a metaphase where you got these X-shaped chromosomes. This is not, this is an example of a bias, because chromosomes are typically not hooked together like this, except at this phase in the cell cycle. Okay, normally these chromosomes would be two different pieces. Now they're hooked together. The center of the X is the centromere, and the centromere is a centromere on both chromosomes, and they join together to make the pair like this. You'll notice that they use the terminology P-arm and Q-arm. They use that to identify parts of the chromosome. And then you have the sister chromatids. And then the reason I selected this particular photograph is that it identifies the telomeres, the euchromatin, which is light color, and the heterochromatin, which is dark color. Now, the heterochromatin, you'll notice, is out near the telomeres and also in the centromere, around the centromere. There's several reasons for that. One is when you start pulling these chromosomes apart, they are subject to mechanical stress, just like any other system. And there's going to be drag as you pull that through, and it's probably a good idea to have this heterochromatin, which tends to be more strongly reinforced near the point where the drag is, the force is being applied. Probably would have more breaks in these chromosomes if you did not have that. As a matter of fact, we talked about paracentromeric inversions, and I'm fairly confident that a lot of those occur because of the physical drag that is applied, and it breaks just outside the paracentrometric heterochromatin, because that's actually going to be one of the weakest part, mechanically weakest part of this process. All right. So if they break on both sides, then you can have an inversion, and pretty much otherwise none the worse for wear. Now, the telomeres are the ends of the chromosomes, and I'll talk about that for a second. Let me put this down. The telomeres, anybody who's ever worked with rope or whatnot realizes when you get to the end of it, it tends to fray. So the telomeres sort of have two functions. One, they tie the end of the rope together so it doesn't fray. It's like that, you go cut a rope or a piece of string, you get all these little strings come out. That keeps that all welted together so it doesn't unravel. The other thing it does is that when you are reproducing, when you do DNA replication, the proteins that make that happen have to have something to grab onto. And at the end of the DNA, it doesn't hook on properly. And so you can't replicate the last little part of the DNA. So the telomeres are actually out there as sacrificial bits, such that as you go through, every time you go through a generation of the cell, that is a cell cycle, you lose a little piece off the end. And this is, obviously you can't incorporate much in terms of the codes for proteins and whatnot, or you'd lose them. And so as you chew down the telomeres, you basically age the cell. Or as you age the cell, going in steps of cell cycles, you lose part of that. All right. Now, one thing I forgot to mention is these are not my graphics. I stole these off of the internet through Bing, and basically it's probably all copyrighted material. I thank the authors. One thing I will say is that I looked at a lot of different diagrams to pick these, and if you happen to have your video, your diagrams on my videos, it's because I think they're very good. Okay, so and if you want to sue me, fine. I have no money. Go right ahead. But I would like to acknowledge all the people who produce these things. People spend a lot of time. I really respect the graphics people that do this stuff. Now, if we look a little more detail at this, and I guess I should have done something more rigid. All right, you'll notice the scale of that big X-looking chromosome as 1,400 nanometers. All right. All right. Clearly, we can see that with a microscope. All right, because that's well above 300 nanometers. If you take one strand of that, it's a supercoiled strand. It's, we're looking at it at a scale of about 700 nanometers. If you look at the actual strand, it's more like 300 nanometers. You'll notice we've gone below the threshold, the 350 nanometers, the wavelength of light that we can see. And if you continue down, you look at one section out of that strand, you'll notice it's a coil. Now you're starting to see something you probably didn't expect. It's not just raw DNA. There's some sort of substance in there. And if you go looking at an individual strand of DNA, you will find that at the 11 nanometer scale, you have these what are called nucleosomes, and the DNA is wrapped around the nucleosomes. Now, the reason we do that, and then the reason that we supercoil the DNA once we've had it wrapped around the nucleosomes, is that it's just, the DNA is a very thin line. In the top, notice there, you'll see it's about 2 nanometers. And it's much, much longer than that. It gets totally tangled up. It's all compacted into the nucleus of the cell, if you happen to be a eukaryote. And it would be a total mess to try to deal with that. It's just like an entangled rope or an entangled garden hose or an entangled roll of yarn. But just like we do by putting string on a spool or a bobbin, these nucleosomes act as little bobbins, and we can wrap our DNA around the nucleosome. There's 2 wraps around each nucleosome, and there's a little connector section of DNA between the nucleosomes. And the nucleosomes are made up, the core of that is made up of protein, and I'll talk about that more in just a second. Keep in mind the scale we're talking about for these things. This diagram, I just want to look at the top of this. And basically, this is a little bit better picture of the nucleosomes. You'll see the DNA wrapping around the nucleosomes. You'll see the, you'll notice that there's one piece of DNA that, not DNA, it's actually a histone. These are histones that make up the nucleosome. The proteins are called histones. And there's one histone that wraps across the 2 strands. That's called histone 1. And then if you look carefully at those nucleosomes, you'll notice that there's an equatorial divide, and then that the spherical shape they have there is divided into 8 pieces. It's the top and the bottom, and then the top is divided into 4 pieces, the bottom is divided into 4 pieces, and that's the way it goes along. And that gives you an idea of sort of the structure of the nucleosomes and how that relates to the structure of DNA. All right. Now, if you go a bit more detailed, nucleosomes, you'll notice that there's turns out there's, I guess there's 5 actually different types of histones. Histone 1 is the one that was locking everything together. The ones that form the core of the nucleosome, there's histone 2A and 2B, and then there's histone 3 and histone 4. So you get, for example, the top of the upper hemisphere, you have a histone 2A and 2B, and you have histone 3 and histone 4 make up the top, and then you have the same thing in the bottom, and that gives you 8 histones making up the nucleosome. And the section of DNA between the nucleosomes is called the linker DNA. All All right. This is a graphic which, you know, is easy to, you know, look at. There you see histone 1 holding everything together, and then you see the histones that make the core of the nucleosome, and there's the DNA coming in and out of it. And down at the bottom of this picture, the second photograph I've put on this page, is you have a another detail comes to light. There are tails coming out of it. Remember, the histones are actually proteins. They're not spheres like that. They're proteins, and they have a three-dimensional shape. And it turns out each one of them has a tail that comes out of the center of this histone. And that's very important for how the histones work and how this whole process works together. Let me step into that and give you more discussion about it. Remember, I'm a chemist, and I look at things from a chemical standpoint usually. It turns out that the DNA, I'm a little bit out of focus. I'm not sure I'm in focus yet or not. Hit the autofocus. Maybe I'm autofocused. Okay, whatever. Maybe I'll come into focus eventually. I'm a little bit out of focus all the time. There we go. I think it caught me. I don't know. Maybe I should come closer. I was holding that all up very close to the camera. In any event, there's two activities that go on with DNA. The whole discussion that most people focus on is the double helix. But the double helix is modified in terms of methylation of CpG groups to provide an epigenetic code on the DNA. And then, I can't figure out why I'm not getting in focus here. Maybe I'll just take that off. Why is that up there? Ooh, suddenly very dark. Sorry, playing around with the video here. Oh my goodness. There we go. Why am I so dark? Now I didn't turn the lights off. I'm not sure what's going on. Turn up the brightness. There we go. There, I'm back. I'm even in focus now. Like I said, these are lectures. These are not productions. I do not edit the videos. Going back, not only do you have this epigenetic marks, these methylations on the DNA, the histones are also marked. Let me focus on that a minute. Out on the chain, that tail that you were looking at of the histones, there are amino groups, not necessarily the amide groups, but the amino groups that may be sticking off the edge. And those amino groups are basic, meaning what they tend to do is pull a proton off of a water molecule. When they do that, it's an ammonium, which is a positively charged system. Now, one thing people totally feel about DNA is that it is charged. All those phosphate groups on DNA have negative charges. And so DNA is always accompanied by a bunch of cations, positively charged salt, you know, like potassium or sodium. As a matter of fact, potassium seems to concentrate in the nucleus around the DNA, which has some interesting effects down the road I won't get into. But the potassium and I guess sodium ions in particular are going to be around the DNA because the DNA is negatively charged, and these things are the counter charge to them. And water molecules are there as well. Now, when that amino acid, when that amine that hangs off the amino acid of the backbone is protonated, it has a positive charge. When it has a positive charge, it's going to repel cations, and it's going to attract anions, negatively charged ions. All right. I don't know exactly how this works in terms of the biology, but I do know about the chemistry. Okay, now as you add methyl groups to an amine, these are like single an alkylamine. It's got one alkyl group and two protons on this amine. When you add a proton to it, it has a positive charge. Now, if you that proton that's a very labile system. In other words, that proton comes on and off very, very quickly and very, very reversibly. As you put a methyl group onto that, you make the amine more basic. So it wants to have more protons associated with it, or have a proton associated with a greater percentage of the time. It's still very labile. If you put two methyl groups on that amine, now you have a tertiary amine, that is to say 3 alkyl groups hooked to an amine, which makes it even more basic. It will keep a proton longer, or will tend to be protonated in aqueous solution, which makes it positively charged, which repels the positive charges, attracts the negative charges. And if you go one more step, if you put another methyl group, you can have 3 methyl groups on this alkylamine, which makes it a tetramethyl or trimethyl ammonium salt. Now it's got a positive charge. It doesn't matter what the pH of the solution is, it's always going to be positively charged. And it is pretty irreversible to take those methyl groups off of there. You can take them off. There are biological ways to do that, but it's not instantaneous. It's not a labile system anymore. It is a fixed positive charge on that histone. Now they're going the reverse. If you took and put an acetyl group, that's like acetic acid, the acetyl group off acetic acid, if you put that on that amino acid, that side chain off the amino acid, that amine now becomes less basic. In fact, it becomes sort of just neutral. Doesn't want to pick up or lose protons particularly. Okay. So as you do that, then you change sort of in the other direction. These are epigenetic marks on the nucleosome. Now, the whole process then of whether or not something gets transcribed is really two steps. First, the nucleosome has to unravel and let the free chain of DNA loose so that it's available for transcription. And then you have to have a signal on the DNA chain, the epigenetic mark of the DNA chain, that says, hey, come and transcribe this section, and a transcriptase will come and transcribe that section. So there's really two things. If the DNA is totally wrapped around these nucleosomes, it doesn't get transcribed. But if it gets out there, it doesn't necessarily get transcribed either, unless it has a signal that says, come transcribe me. Now, there's been a little bit of misconception, which was common until about 20 years ago, that DNA that is what we call heterochromatin DNA, that never gets transcribed. That's not true. Virtually everything in the DNA gets transcribed, but it's not transcribed readily. It only gets transcribed under special circumstances, because it has to unroll, come out as a double strand DNA of the DNA, the double helix, and that is what gets transcribed. So if it's all balled up around a nucleosome, it's not going to be available for transcription, even if it looks like it would normally get transcribed. All right. So this is a point that you need to remember as we go forward, because a lot of my hypotheses are going to depend upon the behavior of heterochromatin versus euchromatin. All right. Now, so I think I've sort of gone through the pieces that are available on the internet, and I looked high and low for a cell cycle that I could use to talk to you about this, but I couldn't find one, so I had to do my own. And this is in part because of the bias I mentioned earlier, which is against, it's for things you can see through the microscope and against things you can't see, basically. They get ignored. So give me a second. I'm going to pull up another graphic that I created. Again, I'm not an artist, and I'm not doing this to impress you. I'm doing this to convince or to educate you. Let's hope this is more or less in focus. Okay, what you see in most pictures that you'll find in textbooks and about the cell cycle is this step right down here, the prophase, metaphase, anaphase, telophase, and cytokinesis. And the reason that's high visibility in the textbooks and whatnot is because that's what's high visibility under a light microscope. That's the bias. Actually, that's just a repetitive step which is pretty much all mechanical and has almost nothing to do with the important parts of the cell, the DNA. Over here, if we start looking beyond what we can see in the microscope, we have what is actually the important thing. The cell has a certain biological function. That biological function is determined by the proteins that are transcribed, and this is where that happens, over here in what we call G0. This is not even visible through the light microscope. This was called the resting cell, which I think is absolutely contrary. This is where the cell is working. This is where the cell is doing what it's supposed to be doing. This is what makes a muscle cell, a bone cell, a kidney cell, a liver cell, etc. Whatever cell it is, that's where the actions is. All right. In this phase, the DNA is deployed. It's inside the nucleus of a eukaryote. It is the DNA strands are out there. They're being actively transcribed, and then all the other things that happen with the transcription and translation of the DNA is happening. But this is all happening in the nucleus. And there's a lot of damage that occurs because every time you're transcribing and moving these cells around, the strands of DNA around, they're very thin, they're very fragile, and they get broken. Things happen, okay? And so there's all sorts of DNA repair mechanisms going on out there during this process, okay? But those processes, those DNA repairs cannot keep up with all the things that happen. And sooner or later, the cell is either going to be called for developmental purposes or for just, hey, I've got damage out here, I need to reproduce this, need to repair it all. It's going to try to bring that all back together and go through the entire reproductive cycle here of the cell. Okay? So what happens is all this deployed DNA, all this single-stranded DNA that's not wrapped on nucleosomes and whatnot, is going to start being drawn back together. And it comes through here, and it's basically checked. It's evaluated for major damages. For example, is there a loop in the DNA? Is there something sticking out the side of the DNA? Is there is a single strand broken in the DNA? Something like that. And all of that needs to be repaired. And there's systems that check this. The ATM protein, among others, check various things about the DNA and feed that forward as a signal that I call stress. The cells are subject to a variety of types of stresses. This is what I call a genetic stress or genomic stress. It's damage to the DNA. But the cell also has stress, for example, metabolic stresses. Does it have glucose? Does it have oxygen? Those types of things. Does it have all the things it needs to do its job? Those are related to stresses as well. And then you have mechanical stress, like is it under pressure? Is it the temperature too hot? Is the temperature too low, etc. All of these stresses get integrated together by the BCL2, not BCL, the P53 molecule, the P53 protein. This acts as an integrator of all these different stress signals here, determining its activity. Now you have sort of a scale set up by two sets of proteins. You have the BCL2 proteins, which are basically resist any sort of adverse action to the cell, and you have the BAX proteins, which are a complementary family, which basically say, hey, we have a big problem here and we need to do something about it. So depending upon where the threshold is set by the availability, the relative availability of these two families of proteins, you have the signal coming in from P53, and if it gets high enough, it will tell us to slow down, okay? And basically the P21 protein is activated and slows down the cell cycle. Now, why would we slow down the cell cycle? Well, there's DNA repair going on as the DNA is being recovered, and if we slow it down, we give it more time for the DNA to be repaired, such that that should relieve the stress that's coming in here. If the DNA gets repaired, it doesn't show up as stress. If it's not repaired, it shows up as stress, all right? So if we can slow down the cell cycle and hopefully reduce stress. But if we go over a certain threshold, then the BAX molecules, the pro-apoptotic molecules, kick in through P27 and activate something called the caspase cascade. Caspases are proteases which basically chop up all the proteins in the cell. It's a cascade of different proteases, and one generation creates another generation, and they go to attack all the proteins of the cell. And most people call this cell suicide. I basically call it a recycling process. Basically, the proteins are chopped into pieces which can be turned into nutrients for other cells. Okay, and so this is a nutrient forming process. It's a recycling process, not really suicide and waste necessarily. But this would take this cell out of action, obviously, if all the proteins get chopped up here. All right. Now, if we make it through that step, if we're not stopped here and not destroyed here, we can make it through to the synthesis process. All right. In the synthesis process, we have something else happen. Now, you can have DNA that looks totally normal from a mechanical standpoint. In other words, if it's no loops, no breaks, no problems like that, no things sticking out the side, it will look mechanically like DNA. You would not know that it is not the right DNA. It may not have the right codes because there may be missing information. You may have lost sections. You may have deletions. Very unlikely you have additions, but you may well have deletions in the DNA. I think what happens is these two strands get compared, and where you have a break, a missing piece, it says, oh, we need to replace that, and it basically breaks the DNA, breaks that strand, and reproduces it using the unbroken strand as the template. It's called homogeneous repair, and so you have homogeneous DNA repair going on, basically doing the synthesis process. As a matter of fact, synthesis and DNA repair are almost one and the same, very similar processes. Well, once you get through the DNA repair and synthesis activity, you go into what we call G2. It's what's called G2. Well, what's happening in G2? Well, you just took the strands of DNA and replicated them. And now you have the original strand. If you go back over here, you know what was going on is that the original strand was synthesizing specific or coding to synthesize specific proteins. And that's because of the epigenetic marks that were on it. When you added that new strand, when you created the new strand of base pairs, you did not create the DNA markings. You did not put the epigenetic markings in there. What's happening here is you're getting the epigenetic markings caught up. Now, typically, the markings that are already there will be used as a code to tell where to put the new marks. In other words, the marks that are on the parent strand are going to be transferred or reproduced or replicated on the daughter strand. That's pretty straightforward to do that. And so we go through G2 and we're basically catching up the epigenetic markings, replicating the markings that are on the parent strand onto the daughter strand. Okay, now we go into what's normally called mitosis, the cell membrane, the nuclear membrane breaks down, and now we've got to take that DNA and we've got to coil it, and we've got to repackage it and back as a normal packaged DNA. And so it's got to be coiled around these nucleosomes. Nucleosomes have to form, and those nucleosomes have to carry the proper marks, which are guided by the epigenetic marks on the DNA. Tells what marks to put on the nucleosomes. So that's all happening in this part. Then we go through the simple separation, the mechanical separation of the two cells, of the two sets of DNA, and then ultimately the two cells split out. You've got the daughter cell and the original cell. All right. So we typically would have two new cells here. They might be identical or they might not. That's called differentiation. All right. So what we're going to talk about now is differentiation and development. Favorite drink here. Okay. How do I look at differentiation and development? Well, going back to our single cell, our fertilized egg, it's presumably got everything we need to make an adult. It's going to go through a series of steps to get there, but it's going to get there. I would argue, based on what we've seen, that this cell does not need any external help. It doesn't depend upon gravity. It doesn't depend upon sunlight. You can have people in Arctic climates. You can have people in tropical climates. You may have people who have starvation diets. You may have people who have plenty of food diets. You may have people have high protein, high fat, whatever type diets. Some people will be inclined to have very physical labor, lots of energetic activities. Other people may be almost totally sedentary. But the cell still goes through the process. Identical twins stay identical. They even stay identical when they develop outside the mother. Okay? Even after birth, they're still developing, and they continue to develop. Although they may be separated at birth, raised in totally different environments, different family environments, etc. So, how do I conceptualize this? I view myself as think about me. I started out historically, my family started with the biological Adam and Eve over in Africa someplace, and came along, went through a number of changes and generations. Every generation's different, and eventually got to me. And so I am sort of a unit in this population, this worldwide population, this national population. And I am what I am because of my history. But the nature of the entire organization, that is to say, all of humanity, depends upon the development of the whole species, not just me. I'm just like one cell in a big organization, big family of humans. That's sort of the same thing that's happening with cells, what we call development. Now I created a little graphic here. This one I did do. It's not very good, but it is me. Sort of the development of an organism. We start off with a stem cell, what we call a totipotent stem cell. It has the potency to make the whole organism again. And that can divide asymmetrically to produce progenitor cells and give you back the original stem cell. And you can have a differentiation into new stem cells. And ultimately, you can end up with the ones here in yellow are the terminally differentiated cells, and they just go through simple proliferation. They just make cells, new cells that are just like themselves. Okay, but that's sort of the step. You have stem cells and progenitor cells, which can make new stem cells or other types of cells, and then you have terminally differentiated cells, which can't do anything except make copies of themselves. All right. So, I'm out of focus again. Every time I do things, I get out of focus. Maybe I'll get back in focus. Maybe I have to get closer and then get further back. Yes, it follows me back. I love it. Okay. So we're getting to the serious stuff now. Okay, pretty much everything I've talked about so far is just replication of other people's work and general ideas. The important thing to note is that the DNA, with its epigenetic marks, both on the cell, on the DNA, and on the histones, determine what the cell does. All right. Now, when I have looked into this topic, if you go into PubMed and you look up asymmetric division or differentiation, stuff like that, what you will find is that I think every paper focuses on the cytochrome, interestingly enough. And we talk about external and internal influences, which provide a polarization. And this polarization that they're talking about is happening in the cytoplasm, because that's what they can see through the light microscope. They can see these polarizations occurring, and they say, well, it's being driven either by the fact that we have this cell in contact with another cell and we're getting signals that way, or maybe it's internal signals, but it's still somehow or other in the cytoplasm, and that's what's driving the cell to do this stuff. If you've watched earlier videos, I'm real down on and we're talking Evo Devo here, I'm real down on the idea of chemical gradients. They're just not stable enough to do what people think, some people claim they do. You cannot have a complex organism like myself that is reproduced in detail regardless of my environment, regardless of where I live, regardless of what I eat, etc. Could not come out this way by chemical gradients. It just wouldn't work. Chemical gradients are not strong enough, not stable enough, not reproducible enough to do this. All right. Similarly, I have the same argument against this idea of external stimuli causing differentiation, causing asymmetric division. Why? Because you're not just cells are not just nice spheres. They are all sorts of twisty, turny things, and they contact each other in many different ways. And you're not in contact with just one other cell. You're in contact with perhaps dozens of different cells. And the idea that this would all work together to provide you with a consistent signal to on one side to say this is going to be division doesn't make sense to me. Also, it's crazy to me. Why are we talking about the cytoplasm when in fact what makes the cell what it is the chromosomes and the epigenetic marks on the chromosomes? There's almost, in the most recent review I've read, they mentioned chromosomes, but they didn't really go into epigenetics and why one set of chromosomes is different from the other. Let's go back, mention one other factor that gets into this, the cloning. If you know, the first thing that got cloned was a sheep. I think Dolly was the name of the sheep. Took a cell from the mammary tissue of Dolly, or from the mother of Dolly, I guess, regenerated the totipotent stem cell, implanted that in another sheep, and lo and behold, we developed a whole new sheep that was identical to the first sheep, except the telomeres were shorter because the mother sheep had been around a while and its telomeres had gotten shot back. Everything needed to make the new sheep, which I think was Dolly, was in that cell. It was not voodoo transferred. So I focus on the chromosomes, and I don't think it has much to do with what it's in contact with. You may be familiar with this, what are they called? Where you get a tissue that's growing in the wrong place. What is that called? Can't think of the word right now. They're, typically they're like teeth and other things that will be like growing in your leg or something. Obviously, that environment in which that development occurred was not in the right place. It just happened to be where those cells were. And so you can have these processes occurring where you get development in the wrong place if the wrong cells end up in the wrong place, or if the wrong cell is in the wrong generation in the wrong place. Okay, so with no further ado, this is the part I'm proud of. This is the new and different thing. Let's see if I've got it. I'll probably start the wrong end. Okay. This gets unwieldy, but we'll go through it. All right. In your initial cell, whether it be a stem cell or a non-stem cell, I'm trying to keep myself so I can see what's going on here and keep you in focus and keep me in focus. Okay, this is a representation of the genetics of a normal cell. This cell has the epigenetic marks are these X's, and those A's represent the coils of the DNA, okay, the base pairs of the DNA. I'm really focused on the epigenetic part. The other part is fixed. All right, so if you have a DNA synthesis process and then you end up with two sets of chromosomes in early G2 phase that are going to look like this. You're going to have one set, the parent set, is going to look like that. It's going to have the same marks. The marks haven't changed, the epigenetic marks. And one strand is not going to have epigenetic marks because it was just synthesized. Well, in G2, you would normally reproduce the marks that were on the parent strand. But suppose you had some renegade RNA, non-coding RNA, that directed where those marks should go. And so this non-coding RNA goes out here and causes those epigenetic marks to show up in the wrong places. You'll notice that they're in different places now. All right. So in the yellow strand with the yellow X's, you have the epigenetic marks of a new strand, a differentiated strand, and you have the original strand. Now you go through this mitosis, and some of you probably say, I understand where we're going now. Basically, if you go through a normal mitosis, you end up with two cells that have asymmetrically marked DNA. I believe this is where we start seeing the polarization in the cytoplasm, because now we've got a nuclei that are polarized already. They have two different sets of chromosomes. Okay, at this stage there are two cells with different epigenetic marks on the chromosomes, on the two strands of one chromosome. Now, if these two cells don't hang around, but if they immediately go through a second mitosis, a second cell cycle, normal cell cycle, and a second mitosis, here's what you're going to have. You're going to have two new cells, one of which looks just like the original parent cell because it has the same epigenetic marks as the parent cell. Gee whiz, the stem cell is back. And then you've got another cell that is completely different epigenetic marks. Okay, now we've got a new cell, a differentiated cell. Now, that stem cell didn't need any sort of external explanation to let it know that it's a stem cell. It's a stem cell because it has the right epigenetic marks. The new cell is whatever it is because it has a different set of epigenetic marks. We didn't have to convince it. We didn't have to move it. We didn't have to identify it any other way. It is what it is because that's what it is. Its identity is determined by its epigenetic marks. We didn't have to have polarization in the cytoplasm to make that happen. Okay, the polarization in the cytoplasm, I believe, was driven by what happened in the genome. And the genome went through a two-step process guided by non-coding RNA that caused the epigenetic marks to change. It's very frustrating to me to read these discussions of asymmetric division, and they never explain how the epigenetic marks changed. They talk about the polarization of the cell, they talk about the polarization of the cytoplasm, and they never explain how you actually change the DNA. Why the new cell functions differently than the original cell. It functions differently because the epigenetic marks on the DNA and the correspondingly nucleosomal marks are different than the parent cell. That's differentiation. The fact that you separated them is just a routine process. It's what you can see under the light microscope, but it's not the fundamentally important process. All right. So that's my story. I've gone just about this. This is the second time back to back I have run through this because this time I do have the camera on, and hopefully I'm setting you up to explain to you how the Master Development Plan, the Master Development Program works. All right. I like everybody else, I tend to use the term plan. I don't want to use the term plan for the reasons I explained in a previous video. Plan means like a picture, like a plan of a building. With that plan of the building, you can't, if I give you the tools and the plan, you can't necessarily do it. You need the program of how you took the tools and raw materials and built the building. That's not in the plan, that's a program. And so that's why I want to use the term program, Master Development Program. And that's a hypothesis which I've been working on. Actually, I published it over 10, I think it's 10 years ago or more. And this, what I just went through here on the differentiation was the insight that I had that would make it work. And the point that flashed out of me after I made this work was I can also explain evolution through this. Evolution takes some more talk and discussion. I'll do that talk and discussion in other videos. I am very close to getting to the point of revealing to you the Master Development Program hypothesis, which you can go read. There's a publication that's listed on PubMed. You can find it in there with my name and Master Development Program. And you can also, I've got a book published on Amazon.com that you can go read. But you know, I think this medium has a broader distribution and is more likely to bring people in, and also get to sort of emphasize the things that I think are important more than the books perhaps do. But the books are more comprehensive. There are literally hundreds of citations in these books. All right. It's not just me spitting this stuff off the top of my head. I give you the citations in the books that will support the comments and the arguments that I make here. I'm giving this to you as a lecture, as I would lecture students who I would tell to go read that chapter in the book to get all the details. I'm not focusing heavily on all the enzymes and proteins and all that stuff. Those are details which are helpful at a biochemical level to explain what's going on. But the concept, you don't need that. You just need to understand how this gentleman went through the steps. if you have some other, if you see a fatal flaw in my work, I'd love to hear about it. I'm a open-minded. I consider myself to be a scientist, and I love to hear other people comment on my work, positively or negatively. There's a comment section you can hit for this video. There are other videos I've got out there that you can also use for this. As soon as I get off here, I am going to upload this to YouTube, and it'll be available. Today is the what is it? It's the 16th of February. And so sometime before midnight on the 16th of February 2022, this video would be published on YouTube, God willing, and the creek don't rise. Have a good day. Thank you.